In this video, we are going to go through the specific steps in DNA replication. And then we're going to conclude by looking at what happens when there are any errors during this process and how they are fixed. There are three basic steps when it comes to DNA replication. They include initiation stage, elongation stage, followed by termination stage. The following figure expresses what happens overall during the process of DNA replication. So we are going to start DNA replication by having a template DNA. This can also be considered the parental DNA, is in a way the DNA of interest that we're trying to make more of. During the step of initiation, what we're going to have is the separation of the two strands. Then, during the process of elongation, there are two major events that are going to be occurring. One is that we are going to have free nucleotides base pair with each of the strands that were present in the template DNA. And then we're going to have polymerases making those nucleotides be linked to one another through their backbone. At the end of the day, we need to terminate the process of replication, and the result is that now if I make the backbone of the newly synthesized strands black, by the end of DNA replication, we're going to have two molecules of DNA composed of one parental strand or template strand, base pairing with one newly synthesized strand. Let's look at these events in detail. So, the following steps of initiation, elongation, and termination are going to be specific for DNA replication in prokaryotes. So I want to make sure that you guys understand that the following steps are for DNA replication in prokaryotes. So in bacterial cells. So in bacterial cells, the process of replication is initiated by having the initiator complex DNA A bind the DNA. As you can see, there's going to be two specific regions that are going to be interacting with this DNA A complex. DNA A is going to be interacting with a 9BP repeat and also with a 13BP repeat. BP stands for base pairs. And understand that these repeats are going to be AT-rich areas. Remember, adenine and thymine are going to be two nitrogenous bases that are going to have two hydrogen bonding events. So it is hypothesized that the reason why these areas are AT-rich is because they contain less hydrogen bonding events when compared to GC regions. In the second step in the initiation stage, we're going to have DNA helicates bind to the parental DNA and unwind it, meaning that it's going to separate the two strands. Once the single strands are separated or are generated, understand that they need to be stabilized so nucleases do not hydrolyze them. And even though they are not illustrated, we have single strand binding proteins that are going to be binding to the single stranded regions. in the last part of the initiation step, we're going to have the enzyme called primase attached to the DNA and synthesize a short RNA primer. This short RNA primer is going to provide the framework for the elongation of the new DNA strand that is going to be formed. Now, during the elongation process of DNA replication, this one is a little bit more complex, and I want to take the time to explain it in detail. Understand that we are going to have DNA polymerase 3 extend the DNA primer that was made by primase. So, one of the things that I want to point out is that when DNA polymerase 3 is extending these newly synthesized strands of DNA, so DNA polymerase 3 is going to synthesize the DNA in the 5' to 3' direction. Now, remember, during this process, we have, and I'm going to highlight it in red, the replication fork separating the two strands of DNA. And at the replication fork, remember that there's enzymes that are allowing that to happen, and helicase is there, as we learned in the previous slide. So in the replication fork, remember, that's where our parental DNA, template DNA is unzipping. Now, remember, DNA polymerase is going to be synthesizing this new DNA strands in the 5' to 3' direction. But let's keep in mind that DNA is double-stranded and it is anti-parallel. That means that we're going to have one of those templates DNA run in the 3' to 5' direction towards the replication fork, and its anti-parallel strand is going to be running in the opposite direction. And in this case, I'm going to highlight the anti-parallel strand in green. Now, when this template from the parental strand, it is utilized, understand that the one that is highlighted in yellow, the one that has the 3' to 5' orientation, that is going to be utilized as a template to synthesize the leading strand. And the leading strand, we are going to be forming it by forming base pairing events with that 3' to 5' orientation of the parental DNA, and is going to be made continuously. So if I highlight this leading strand in magenta, the leading strand is going to be made continuously towards the replication fork. And that's what we are observing. So this is going to continue to go this way as it is opening up. Now, understand that in the case of the other strand that is being formed, because we need to utilize the template in the 3' to 5' direction, that is going to be made in small pieces, okay? And those small pieces, which I'm highlighting in purple, they're called Okazaki fragments. So these Okazaki fragments, as you can see, since they're made in pieces, they are not made continuously. In the next step of the reaction, we have DNA polymerase 1 remove the DNA primers. And remember, there are RNA primers on each one of the strands. And that DNA polymerase 1 is going to fill the gap with new DNA. In the last step of the reaction, in the elongation process, we're going to have DNA ligase join the ends of the DNA fragments together and understand that those fragments that were made by the Okazaki fragments are what we called overall the lagging strand. And remember that this lagging strand is going to be made discontinuously. During the last stage of DNA replication in prokaryotes, understand that what happens overall is that the two replication forks are going to meet at the opposite side of the replication fork. Then, around this region, we are going to have termination sites, and they are not shown in the figure that we have on the left. But these termination sites are what are going to be arresting the movement of the forks. At the end, understand that once this replication has been completed, what we're going to have is that the molecules that have been made of the circular DNA are going to be catenated. And catenated simply means that they are interlinked. Then we're going to have topoisomerase enzymes going to separate the two newly synthesized molecules. So even though they are illustrated in the figure on the left, separated, understand that after the whole process of DNA replication in prokaryotes, we're going to have two circular DNA structures composed of one parental strand that is base pair with the new DNA strand. Understand that DNA replication in eukaryotes is very complicated because remember that it is regulated by the cell cycle. I wanted to illustrate the complexity of the DNA replication in eukaryotes by illustrating this slide. In here, we have a figure that talks about all the different complexes that have to come together in order to start the replication process. The take-home message of this slide is that DNA replication in eukaryotes is more complex than in prokaryotes because there are many proteins involved, there are different replication bubbles in the whole system, and lastly, it is regulated by the cell cycle. Do not memorize the specifics when it comes to DNA replication in eukaryotes. Understand that DNA replication in prokaryotes and eukaryotes to date, it is known that is a little bit more detailed than what I presented to you guys. In total, both replication processes are composed of 5 polymerases. And as you can see, we have outlined what are the functions of those polymerases. Now, the polymerases that are involved in prokaryotes understand that they also are exonucleases, while the polymerases in eukaryotes are not exonucleases. The definition of an exonuclease is going to be enzymes that remove successive nucleotides from the end of nucleic acids. In addition, remember that there are several origins of replication in eukaryotes, while we only have one origin of replication in prokaryotes. The Okazaki fragments for prokaryotes are longer than the ones in eukaryotes. And lastly, understand that when it comes to the proteins that are complexing with DNA, we don't have any proteins complex to prokaryotic DNA, while we have histones, which are complex to DNA in the case of eukaryotes. So, when this whole process of DNA replication is occurring, understand that the process has to undergo proofreading and mismatch repair. What does this mean? This is kind of similar when you're writing a paper. After you write a paper, you need to proofread the content of your paper, and you need to repair any type of sentence that doesn't make any sense. In the process of DNA proofreading, understand that it may be that an incorrect base is going to be incorporated into the growing chain. The DNA polymerase is going to be immediately removing that incorrect base, and is going to replace it with the correct base before it continues with the replication process. Now, if for some reason the polymerase does not fix this, then in the case of a mismatch repair, understand that what happens is that we're going to have mismatch repair proteins that are going to remove the mismatch base in some adjacent bases. Then we have the DNA polymerase at the correct bases, and lastly, we are going to have the ligase putting together the new segments that were made.